Okay, so I'm going to give you guys some specific directions and take you a little bit through the lab, which is investigating biological compounds, okay? I did go over this in a Class Connect, but I'm going to go over it a little bit more in detail with you, and you can follow along and work on this lab, okay? So basically, I've gone through maybe just the first 2 pages, kind of the intro to this lab. But the one thing that you need to fill out and you need to open up is these laboratory instructions, okay? Investigating biological compounds. When you open that up, it'll look like such, okay? It says laboratory instructions. We'll come back to this in a moment because we'll be reading through this so that you can finish up this lab, okay? But back to the actual lab where you're going to find this. Again, in the left-hand toolbar, if you just click on that, you'll be accessing this here. So let's kind of go through it. This is a fictitious crime. So basically, and I'm gonna kind of paraphrase most of the stuff. You guys can actually read through this information here, and I hope you do, because you'll see some of that information in the lab quiz that you must take. Okay, but basically, cops come in. Bam! There's a bunch of what they think to be cocaine in this hotel room. They're trying to figure out where this cocaine came from, right? But they don't know that. They know that there are a couple of different areas in the California area that are producing these mass quantities of cocaine. So they're going to try and figure out which of these basically areas are producing that cocaine so that they can target who is responsible for this. Please read a little bit about what it is. Okay, I'm not going to read that right now. You guys can read that. You can pause this and read what is cocaine. Some of you guys probably know some of those side effects, but you'll see probably 1 or 2 questions in the quiz on that as well. Okay, so again, according to the government information, basically some of these organizations are located in the different areas of California: Bakersfield, Los Angeles, and San Diego. So the interesting thing about this is that, and this is true, cocaine is cut with powders, and that means that basically it's diluted. So you have this pure cocaine, this pure drug, that many different people that try to sell these mass quantities of cocaine will actually dilute it, kind of to make it go further, right? Kind of it's a dishonest way of stretching that. So depending on the place, some people cut that cocaine with different substances. So basically it's saying in here, it says the clue to the origin of the oceanside cocaine may rest in identifying the substance used to dilute it. The DEA knows that the different organizations use the following different substances to dilute their cocaine. The San Diego area is using powdered milk to cut that cocaine. Los Angeles is using cornstarch, and Bakersfield is using a fat-soluble form of vitamin C. Okay, all are white powders. By the looks of it, you cannot see what is what, but by doing particular tests, you can determine what it's cut with. Okay, so that's what we're going to do in this lab, because think about it. If we figure out what that cocaine is being cut with, we can figure out the area from which that cocaine has been manufactured or is being, basically not necessarily processed, but sold and distributed from that, whether it's San Diego, Los Angeles, or Bakersfield. Okay, now let's talk about the different tests that can be run. The first test that can be run is the iodine test, which is an indicator for starch. Let me give you some background on starch, and hopefully you guys know this. This is a biological kind of concept from biology. Starch is a long chain of sugar molecules like glucose, okay? So if you have lots of glucose molecules, you end up with a starch. So lots of chains of simple sugars give you a starch, such as pasta, cereal, bread. That's a starch. But a simple sugar would be like glucose, like table sugar. Okay? There's also fructose, which is the sugar out of fruits. There's lactose, which is the sugar out of milk. So no matter what, if you test for a starch, you will test that substance will test positive if it's a simple sugar or if it's a complex sugar, which is a starch. Okay? So when you add iodine to a solution, and I want you guys, I'll read this part in a moment, but when you add iodine to a solution, if there's a positive test, it will turn one of these colors, really dark purple, kind of a blue, or a light purple. Now, iodine is a kind of orangey color in its natural state. So basically, you're going to start by adding iodine solution to a control solution, water. Okay, water is always a good control, right? Because we know water is not going to test positive. We know it's going to do nothing. So if you add iodine to water, it's going to turn like a rusty color, okay? So that's a negative test. Then you're going to add you're going to add iodine to an actual, substance containing starch. So you know it's going to be positive, and this is what you'll see, okay? Then we're going to test this unknown solution and hopefully figure out, hey, is there a starch in there? And maybe that'll help us determine whether or not, this cocaine came from a particular place. By the way, let me just go over these. Powdered milk. So powdered milk could test for possibly sugar, right? Because there's lactose in milk, which is the sugar in milk, okay? And then it also could test for fats, which, powdered milk typically has fat content. Cornstarch, hint, hint, starch, will test positive for starch. And then this other one, that's this, this actually won't have any positive test for any of the tests that we are going to test for, because we're testing for starch, lipids, and proteins, okay? All right. Oh, I'm sorry. Fat soluble. It said it was fat soluble, so it would test positive for that fat. So that's the iodine. Okay. Now let's go on to the next one. Sudan 3. This is a chemical indicator for lipids. So anytime anything that has fat in it, if you were to add this Sudan 3, then basically it's going to test positive by showing this reddish color. Okay. So again, you're going to have to, in your lab, and I'm going to open up your lab guidelines or your lab instructions, I'm sorry, because you need to be able to explain these different tests, okay? So basically, we went through this first part right here, but the procedure says, open the Investigating Biological Compounds Virtual Laboratory, which we'll get to in a second. We're going to do the tutorial together, and then I'll kind of go through some other things for you. But look at these questions here, okay? After you do this in the virtual lab, which we'll do together, it says, why do we say that an iodine solution is an indicator for starch? So just so you guys know, this portion in your course is helping you to answer these questions. So please read through this very specifically, okay? If you need to pause this or if you want to go back to your course and read through this, this talks about why it has a positive test for starch. Okay, so please read through that, as well as the Sudan 3. You need to understand, it's a class of dyes that interact with lipids to cause a color change, and it'll talk a little bit more about that. So when you add Sudan 3, if this substance has fat in it or lipids, it will turn this reddish color. Obviously, if added to water, this is actually a kind of a reddish color already, so it'll just be kind of a lightish red. But if you will know the difference when you have the lipids that are formed, or the lipids that are there. The actual positive test will be kind of a bright red. And then the last test is the Burette reaction. This is an indication for proteins. So basically, if you read through here, it says proteins are made of amino acids, which contain nitrogen. When proteins are exposed to copper ions and a base, the copper ions bind to the unpaired electrons in the nitrogen atoms, which cause a color change. This is called the Burette reaction. So the Burette solution actually is this color, okay? It's this kind of a light blue. But if you put this in with something that is containing a protein, which is, amino acids put together, it's going to turn this violet color, kind of a purplish, okay? So it says to do the Burette test, scientists add solutions of copper and a base to control the water in the protein solution, okay? So basically, this is what's going to happen if you have a positive reaction to that and that protein is present. Okay, so now it's time to analyze the cocaine sample, because obviously they took a sample from that. Let's go and enter the virtual lab. Okay, if you go on to page 14 of this lab, you'll notice that right here, this link will actually take you to the virtual lab, and we will do that together. So again, it says you will test the unknown cocaine sample for starch, protein, and lipids. You'll use the following indicators, okay? But we are going to set up a control. We're going to do the control solution 1, which is the indicator plus water, which is going to give us always a negative test, right? So we can compare it to it. But let's see what a positive test gives us. So we're going to do the indicator plus the known sample, which will give us that positive test. Then we'll test our unknown sample, okay? All right, so click on that. It says you're going to be taken to an external website. I will try to kind of pan around this. Sorry, I got to kind of move that around. You want to do the tutorial first, because that tells you what to do. So pick your color gloves, la 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 la, okay? And it's not hard, you follow the directions in your lab instructions. So notice how basically, it says procedure, sorry, let me scoot down here. Part 1, open this tutorial, view the tutorial, complete the tutorial. When you've finished it, click the home button, reset the lab, and begin the lab and proceed to the sections below. So we're gonna do the tutorial together. Then we'll do the test for starch. The tutorial just kind of lets you know how to utilize kind of this virtual lab, so to speak, okay? All right, so let's start it. So place your mouse over one of the 4 arrows so you can see that you pan that, okay? You can just use that, okay. Click on the gear to show or hide the menu, so I'm just gonna read through these and quickly go through. Click on the resize icon to change to and from full screen, okay? So you can kind of click on that, which I'm not gonna do right now. I don't wanna change it. Click on the lock icon to turn the scrolling off, so you can click on that. Click on the speaker icon to turn off the audio. Drag the bar towards the magnifying glass, so you can make it bigger, obviously, or smaller, okay? And click on the home icon to reset the lab, okay? So always do the tutorials, because that helps you, right? Okay. All right.
Many of these chemicals are harmful and should be handled carefully using gloves and goggles.
Okay, yes.
Remember to use clean pipettes when needed. Do not use a pipette for more than one specific kind of solution, since it will not have been properly cleaned.
Okay, that's a big deal. If you use a pipette that's already been used for something else, you are pretty much negating your lab, okay? Don't do that.
Place a full pipette of water into the water test tube.
All right, so I'm going to pan out just a little bit so we can see everything. And, oops, I want to go over here. So it's kind of telling you what to do. So here's the water. Let's grab some water here. And let's put it in our test tube, okay? Put it back.
Place a full dropper of starch solution into the starch solution test tube.
All right. Starch, put it back.
Add 1 dropper of the iodine solution into the starch solution test tube.
Okay, so let's test. This is going to be the positive test. Remember, we already added starch, so we're going to add iodine. Look it's purple. Yes, that's positive test because we knew it was starch, right?
Add 1 dropper of the iodine solution into the water test tube.
Okay, remember this is going to be our control, so if we add this, nothing should happen, right? So iodine, I told you, is kind of orangey in color.
Observe the color of the starch solution test tube and the water test tube. A dark blue or black color for the starch solution indicates that starch is present.
Okay, so obviously you can see...
You have reached the end of the tutorial. You may now continue with the rest of the lab.
Okay, so let's click on the home because we want to reset this lab. So let's reset that because now everything is clean and reset for that. Sorry, my dog is chewing. Hey, do not chew on my power cord. Hello? Okay, begin the lab. So choose your gloves again. And now we're going to follow the lab instructions, okay? So it says when starch is present, an iodine solution will turn dark blue, dark violet, or even black. Knowing this, you can conduct a procedure, blah, blah, blah. So we already did this. Place the water filled from the pipette into the starch control. Place the starch, okay? So we did all this already. Test for starch by adding iodine to a filled eyedropper to each test tube and record your observations. So obviously your starch and water, it just turned, oops, an orange color. Gosh, can't spell. Orange color, okay? And really, you know, that would be no reaction. But your test for starch, right? What color was that? Yes, it was purple. Okay, purple. That is a positive test, okay? Why do we say that iodine solution is an indicator for starch? I want you to go back to remember where it talked about that, it talked about, you know, how that is an indicator for starch. So go back and read that and answer that question. So part 3 is testing for a protein. It says when a protein is present, a mixture of sodium hydroxide and copper sulfate will turn a very dark color, basically usually blue. Knowing this, you can conduct a procedure using the solutions in the lab. So place water from a filled pipette into the test tube labeled protein control. So we're going to do the same thing. We're going to do the water, then we're also going to do the protein, okay? So we're going to see what happens with that. So let's go back to our lab, okay? All right, start. So we can take water and get some water. Okay, here's the protein control, water. Let's put that back. Let's see, let's kind of move over here. What do I want to use for protein? Well, let's do the sodium hydroxide and the copper sulfate first. Okay, let me pan out here. So remember, sodium hydroxide into the water, and then copper sulfate. Both of those need to go in to test for protein. Okay, so notice that it didn't change in color, it just stayed kind of that blue color, which is the color of this copper sulfate. So that's a negative test. Okay, so we just did that. Now let's do milk, because don't we know that milk has protein in it, right? So let's do some milk. So test for protein. We know milk has protein in it. So let's add the sodium hydroxide to the milk. Now let's add the copper sulfate and let's see what happens. Okay, notice it turned kind of that bluish purple. That's a positive test, and I can scroll in a little bit closer so you guys can see that. Okay, so there are your negative tests with the water and your positive test for the protein. Okay, we can also do the same thing for the lipid. Okay, so I'm going to pan out again. So look, we have lipids, we have vegetable oil, right? So first of all, let's do our water, because this is our control. Okay. And let's do our lipids, because we know that vegetable oil contains fat, right? So let's put that in our control for the positive test. Now remember, we use Sudan 3 to test for lipids. So let's put some in there. Now this is a very slight difference, okay? Because remember, Sudan 3 is already kind of a reddish color, okay? But even though you can't see this, this is kind of a duller red. This is a brighter red. So you have to really kind of look closely. But in person, it's pretty easy to tell. But this is a brighter red. This is kind of just a diluted color of the Sudan 3. So the control, negative. The test for lipid, positive, okay? All right, let's go back to our lab. Because basically you need to fill out your observations, the protein control, the test for protein. And then it says, why do we say that the basic solution containing copper sulfate is an indicator for solution for protein? Remember, that was in the lab kind of guidelines that I read through in your course. So go back and read through that. Then we did the test for lipids. So I kind of did that for, through what I just did really quick. So you guys can fill out your table. It says, why do we say that Sudan 3 is an indicator solution for lipid? Then you want to test the unknown, okay? So I'm not going to go through this entire thing because I really want you guys to work on this a little bit together instead of just watching and, then you're done with it. So testing for the unknown. So go back to this lab, and if you pan over here, okay, you'll notice there's 3 test tubes, okay, for the unknown. This milk portion will come into play in the last question. But if you want to test the unknown, the starch, right, look it up here. You can see if I pan out just a little bit. You have your unknown solution. This is like, say this is the cocaine from that crime scene. They're going to test it because it's unknown. So you would put your unknown in each of these, right? Because we're going to test for each of these. Okay? And then you're going to test for each of those indicators. I'll let you guys do that. So obviously when you, let me scroll down here a little bit. Your starch, you're going to be adding your iodine. Your protein, you're going to be adding both of the sodium hydroxide and the copper sulfate. And the lipid, you're going to be adding the Sudan 3. Okay? Record what happens. You can figure out maybe a little bit more about where that actual unknown source of cocaine came from. Okay, so back to your lab. You can fill those observations out after you do that. And it says, which test tube showed a completely positive response to an indicator? So just answer that for me. And then you're going to analyze your results. What were the results of this test? What were the results of this test evidence that was individualized or class evidence? Remember, class evidence is like classifying something kind of on a broad spectrum. Individualized is pointing to one individual. Okay? I'm not going to say much more than that because I want you guys to think about that. Is this pointing to one individual, or is it pointing to something as a whole? Okay? So think about that. Number 2, here's where you might want to use those milk test tubes. The lipids in the milk will cause the milk to react strongly with Sudan 3, right? We know that there's fat in milk, even if it's powdered. But if you knew that the milk also contains some sugars, how might this lead to testing errors? So remember, milk does contain a type of sugar. I told you guys that in the beginning, lactose. Think about how, and I want you guys to use those 3 test tubes and kind of see, how might that kind of skew your results and what you might have to do further testing. So think about that, okay? That's the end of the lab. Pretty easy, right? I think so. So there you go. If you have questions, you definitely can let me know, but that is it. Now I need to pause this, and I cannot.